Hi friends, and now we'll study significance of mitosis. Growth in organism means an increase in size. It occurs due to increase in the number of cells. Mitosis plays a crucial role in growth by producing new cells that are identical to the original cells. Cell replacement. Many cells are constantly dying in our bodies. For example, the red blood cells and the cells of the walls of intestine and skin, etc. These are replaced by new ones which are exact copies of the older cells. The new cells are formed by mitosis. Regeneration. Some animals can regenerate parts of the body. For this purpose, they form new cells by carrying out mitosis in the cells of remaining parts. Asexual reproduction. Mitosis of means for asexual reproduction. For example, Hydra reproduces asexually by budding. During this process, mitosis forms a mass of cells called bud on the surface of Hydra. Mitosis continues in the cells of the bud and it grows into a new individual. Errors in mitosis. Sometimes the process of mitosis goes wrong. For example, during anaphase of mitosis, the sister chromatids of a chromosome may fail to separate. As a result, one daughter cell receives both sister chromatids and the other will receive none. Chromosomes may also be damaged during mitosis. If the genes that regulate mitosis are mutated, change, the cells continue to divide. Due to this uncontrolled division, masses of cells are formed. These masses are called tumors. If the tumors remain in their original location, they are called benign. If they migrate and invade other tissue, they are called malignant tumors, cancer. It is called metastasis, spreading of disease. Meiosis, it is the type of cell division in which each daughter cell receives half the number of chromosomes as compared to the parent cell. In meiosis, a haploid parent cell divides to produce four haploid daughter cells. Diploid means the cell in which chromosomes are in pairs. Homologous pairs. By haploid means the cell in which with half number of chromosomes, for example, cells with no pairs of chromosomes. Phases of meiosis. Meiosis was discovered in 1876 by a German biologist Oskar Hartwig. Meiosis consists of two divisions, meiosis 1 and meiosis 2. Meiosis 1. In meiosis 1, the homologous chromosomes in a diploid cell separate and so two haploid daughter cells are produced. It is subdivided into prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1. During this stage, chromatin condenses and takes the shape of chromosomes. Each chromosome consists of two sister chromatids because the DNA has already replicated before meiosis. Homologous chromosomes move closer together. They pair up in a process called synapsis. Each pair of homologous chromosomes is called tetrad. Non-sister chromatids of homologous chromosomes become zipped together, forming X-shaped structures called chiastema. Each chiasma is the site of for crossing over exchange of portions of chromosomes between non-sister chromatids. Crossing over leads to recombination of genetic material. Other events of prophase 1 are similar to prophase of mitosis. The nucleoli disappear and nuclear envelope breaks. Centrioles migrate to opposite poles and make spindle fibers to which chromosomes attach. Metaphase 1. The tetrads attach with spindle fibers elegant along the equator. In this way, they form metaphase plate. In the metaphase plate of meiosis 1, two spindle fibers from both poles attached with one pair of homologous chromosomes. Anaphase 1. Each spindle fiber attached with a single chromosome pulled towards the pole. In this way, the paired chromosomes are separated. One chromosome of each pair is pulled towards the one pole and the other towards opposite pole. So two haploid sets of chromosomes are formed. Each chromosome still contains two sister chromatids. Telophase 1. Spindles disappear and a new nuclear envelope is made around each haploid set. Nucleolus also reformed during telophase. That chromosomes uncoil into chromatin. Cytokinesis occurs and two daughter cells are made. Meiosis 1, Meiosis 2. Meiosis 2 closely resembles mitosis and consists of four phases: prophase 1, metaphase 2, prophase 2, metaphase 2, anaphase 2, and telophase 2. In prophase 2, the nucleoli and nuclear envelope disappear and the chromatin condenses. Centrioles move to the poles, forming spindle fibers. During metaphase 2, spindle fibers attach to the kinetochores of chromosomes, aligning them at the cell's equator. In anaphase 2, spindle fibers pull sister chromatids apart towards opposite poles. Finally, in telophase, two chromosomes uncoil back into chromatin. Nuclear envelopes and nucleoles reform and cytokinesis occur. This results in the formation of four daughter cells, each with half number of chromosomes. In animal cells, in reproductive organs undergo mitosis. The daughter cells and called gametes have half the number of chromosomes with no pairs. During sexual reproduction, male and female gametes join to make the first cell zygote of new generation. The original number of chromosomes is restored in zygote. It undergoes mitosis many times and develops into the new animal. In flowering plants, specialized cells in flowers undergo meiosis. The daughter cells called spores have half number of chromosomes. These spores grow into new generation inside the flower. This generation produces gametes by mitosis. The gametes join to make zygote with full sets of chromosomes. The zygote undergoes mitosis and develops into the new plant.